Amplification of DNA, or making lots of copies of DNA, is necessary in order to be able to isolate genes or to characterize them, or to carry out transgenesis or make recombinant protein. Molecular cloning is an important technique for amplification of DNA, that is, making lots of copies of a DNA molecule. And molecular cloning leverages a natural biological process, the process of bacterial cell division, to make copies of DNA. Bacteria have or can have DNA outside of their chromosome, that is, episomal DNA, called plasmids. One such plasmid is the F plasmid, which carries the fertility factor for bacterial conjugation. Now, bacteria will take up these plasmids, which are circular in shape, from the extracellular environment in a process known as transformation. And in molecular cloning, scientists take some DNA fragment that they want to amplify or want to make lots of copies of, and this DNA fragment is introduced into the circular plasmid, and then this circular DNA, the plasmid DNA that carries the fragment that needs to be amplified, is introduced into bacteria using transformation. And then the bacteria are allowed to grow. And bacteria, E. coli, they divide once in every 20 minutes. And therefore, they divide 3 times in 1 hour. That is, they will divide 36 times in 12 hours, which means that we will have 2 to the power 36 times the DNA that we started with in 12 hours, and 2 to the power 36 is approximately 7 billion times. So we will have, if we start with one bacterial cell that contains one plasmid, we will end up with 7 billion molecules of DNA in just a short period of 12 hours. So how do we make these plasmids that contain the fragment of DNA that we are interested in amplifying? So these plasmids, they're called recombinant DNA, or a recombinant plasmid, or a recombinant vector, since they're carrying the fragment that we want to amplify. And they're called recombinant because they are recombining 2 different types of DNA. This fragment might be from the human genome, whereas the plasmid DNA in cyan is bacterial in origin. So we have combined DNA molecules of very different origins into one DNA molecule, and that's why this is called recombinant DNA. And this molecular cloning is also known as recombinant DNA technology. The first step in introducing a DNA fragment into a plasmid or a vector is to actually cut the DNA. And we use, for this purpose, these enzymes called restriction enzymes. And these restriction enzymes evolved as a defense mechanism against viruses for bacteria, that is, bacteriophages. And let's use the example of an enzyme called EcoR1, which is from E. coli, hence the name Eco, and R1 stands for restriction 1. Now this enzyme, what it does is, on all restriction enzymes, what they do is they recognize specific DNA sequences. And EcoR1 recognizes a sequence GAATTC, and this is a double-stranded molecule. So we have CTTAAG, and you will notice that the complementary strand has the same sequence except flipped, and so these restriction sites are palindromes. And what the restriction enzyme does is it breaks the phosphodiester bond between the G and the A on both strands of DNA. So the restriction enzyme recognizes this pattern of sequence GAATTC, and this pattern is called a restriction site. And every restriction enzyme recognizes a different pattern, has a different restriction site. And in E. coli's case, when it recognizes this, it cuts or breaks the phosphodiester bond between the G and the A. And what that means is that we will have a DNA molecule that looks like this. This G will have its 3' hydroxyl. Since the phosphodiester bond between the 3' hydroxyl of this G and the 5' phosphate on the A has been cut, and then we will have the 5' phosphate of the A, and then AATTC. And the other molecule, or the other strand, is going to look like this. You will have the C and TTAAG with the 5' phosphate, and you will have the 3' hydroxyl of the G on this end. And one thing to note is that this G and C will are still base paired because they have their phosphodiester bond with the nucleotides next to them. However, the AATT sequence on both strands are free to move about, since the phosphodiester bond between the G and the A has been broken. And so if you have one piece of DNA with the restriction site and you cut it, you will end up with 2 pieces of DNA. On the other hand, if you have a circular piece of DNA and you cut with the restriction enzyme, you will end up with a linear piece of DNA. Another thing worth pointing out about this DNA molecule where the phosphodiester bonds have been broken by the restriction enzyme is that these two ends, they can still align with each other and form hydrogen bonds. However, these hydrogen bonds can be easily broken since there's no phosphodiester bonds between the G's and the A's, and therefore the two ends will sort of go back and forth between these two conformations, one where they are sticking to each other by hydrogen bonds, and the other conformation where they are separated from each other. And this ability of these overhangs to stick to each other make this a sticky end. These are called sticky ends, and that's a property of the enzyme where if it cuts the phosphodiester bonds in this staggered manner, it creates sticky ends. There are other enzymes such as eco R5 that actually do not have sticky ends. The restriction site is GAT, ATC, and it's palindromic. And eco R5 cuts between the T, N, and A so that there is no overhang and there are no sticky ends. And these kind of sites are called blunt end. So how can we use restriction enzymes to introduce fragments that we are interested in amplifying into circular bacterial plasmids to make recombinant DNA or recombinant vectors? First, we digest the plasmid, and so I have my plasmid in red, and the fragment, the DNA fragment that I want to amplify is here in blue. And both the plasmid as well as the fragment have eco R1 sites in them. In the plasmid, there's one eco R1 site, whereas in the fragment, there's two eco R1 sites on either end. And these sites can be added if they don't already, if the fragment doesn't already have them, they can be added using blunt end cloning. So we will go ahead and separately digest both the plasmid and the fragment is usually called the insert because we're going to insert the fragment into the plasmid, and we can digest both of them with eco R5. And when we do this digestion, what's going to happen is that for the fragment, we're going to lose part of the eco R1 site, and the plasmid is going to become a linear molecule after digestion because of the breakage of the phosphodiester bonds. Next, after digesting the plasmid and the fragment with eco R1, the two molecules or the two DNAs will be mixed together. And since you have sticky ends, what's going to happen is that the DNA fragment is going to come and the overhangs are going to base pair. With the overhangs in the plasmid. And that's going to be true for both ends. Now, even though they are forming hydrogen bonds, as we discussed previously, there are no phosphodiester bonds between the G's and the A's, and therefore the plasmid and the fragment, they're going to go back and forth between these two conformations, where they might be sticking, like over here, or they might not be sticking to each other, like over here. And in order to make sure that the insert stays within the plasmid, we have to make the phosphodiester bond between the G and the A. The G over here has a hydroxyl group on the third carbon, and the A has a phosphate on the fifth carbon, and the same holds for all the G's and the A's. So the G's have hydroxyl groups, and the A's have phosphate groups. And what our objective is to make a phosphodiester bond by carrying out the condensation reaction between the hydroxyl groups on the third carbon of the G's and the phosphates on the fifth carbon of the A's. And this can be done by treating this mixture of the plasmid and the insert with the DNA ligase enzyme, which is going to make a phosphodiester bond between the G's and the A's, and that will ensure that we will have a continuous DNA molecule in which the fragment that we want to amplify has been inserted into the plasmid, into the EcoR1 site. And now, having made these phosphodiester bonds and succeeding in making this recombinant DNA molecule, we can proceed to transform E. coli with this recombinant vector recombinant plasmid in order to amplify this this fragment, the blue fragment that we want lots of copies of. At this point, we run into two problems. The first problem is that not every bacterium, when we transform them, not every bacterium will receive the plasmid. And so there will be some bacteria that have the plasmid when we transform, and others, like this one over here, that don't. And usually the transformation is a rare event, and the vast majority of bacteria are not going to have the plasmid. And so when we try to grow these bacteria, the chances that we will run into a bacterium that actually receive the plasmid is pretty slim. To overcome this problem, what scientists have done is they introduce into the plasmid, or rather the plasmid carries an antibiotic resistance gene, like this ampicillin resistance gene shown in red. What this implies is that all the bacteria that receive the plasmid are ampicillin resistant, whereas the bacterium that does not receive the plasmid is ampicillin sensitive. And so you plate these bacteria, which means you take a solution of these bacteria and dilute it down so that when you spread it out on an agar plate, the individual bacteria are isolated, and then you let the bacteria grow so that you have colonies after about 12 hours. However, all the bacteria that are ampicillin sensitive will die because you include ampicillin in the medium when you are growing these bacteria, and only ampicillin resistant bacteria will survive. Since it's the plasmid that confers ampicillin resistance to the bacterium, any bacterium or any colony that is resistant to ampicillin must be carrying the plasmid. This process of selecting bacteria that carry the plasmid is called selection. And since ampicillin acts negatively, that is, it removes the bacteria that are not desirable, that are not carrying the plasmid, this is called negative selection. A second problem we face is that when we insert the fragment into the plasmid, and that is when we ligate the fragment and the plasmid, not every plasmid molecule will receive a fragment, and some plasmid molecules will recircularize. That is, the EcoR1 sites, they will stick to each other and get ligated without receiving the insert. And we should have another strategy for picking only those bacterial colonies that have that correspond to plasmids that received the insert. One way of achieving this is known as blue-white screening. And the way blue-white screening works is that the vector or the plasmid, in this case we're using an example of the PUC18 vector, it contains the LAC-Z gene under the control of the LAC promoter, and this gene will make the beta-gal enzyme. And when growing these bacteria, you add this compound X-gal to the medium, and beta-gal will cleave X-gal and create a blue color. So any vector or plasmid that has a functional LAC-Z gene will make beta-gal, beta-gal will cleave X-gal and result in a blue colony. However, if you have a successful insertion event and the EcoR1 site, for example, is in the middle of the LAC-Z gene, so if you have a successful insertion event, your fragment of interest, the fragment that you're trying to amplify, gets inserted in the middle of LAC-Z, the LAC-Z gene, and therefore it creates a mutation in the LAC-Z gene that results in a beta-gal that is not functional. And so you have created a mutation where you don't have a functional beta-gal. Now, if you have a successful insertion event, like here, you will not make beta-gal, and there will be no cleavage of X-gal, and the colony will be white. Therefore, we can pick bacteria that have plasmids containing the insert simply by choosing or picking a white colony rather than a blue colony. And therefore, this is again selection, we are selecting for a particular trait in this case, or a feature in this case, whether the insert has been integrated into the plasmid or not. However, since we are choosing the acting upon or choosing the colony that contains a vector, this is positive selection and not negative selection, as in the case of the antibiotic, where you were killing off the bacteria that you did not want. And because of the colors involved, this is known as blue-white screening. And the endpoint of this process is that any one of these white colonies will contain bacteria which have a plasmid in them, and that's because the plate contains ampicillin, and the colony must be ampicillin resistant, and the resistance comes from the plasmid. And since the colony is white, it must contain the insert in the plasmid, since white implies that there is no beta gal, and which means that laxity has been disrupted by our insert. And we would take one of these colonies and then grow it in a large volume of media to amplify the bacteria and therefore amplify our fragment of interest.